start with this surprising, almost unbelievable idea. There is a group of bacteria so common it lives, well, literally everywhere, in the soil, in the water, and yes, probably right now, inside your shower head.
Right. And for almost every creature on Earth, it's completely harmless.
But for a very specific, genetically vulnerable subset of the population, we're talking about certain dog breeds, this ubiquitous organism is a swift and fatal threat.
And that contrast is really the key, isn't it? The fact that the pathogen is everywhere, but the disease is incredibly rare. That's the mystery we're digging into today.
We are taking a deep dive into the Mycobacterium avium complex, or MA. This is part of a larger group called non-tuberculous mycobacteria, or NTMs.
Exactly. And when most people hear mycobacteria, their mind immediately goes to tuberculosis.
Right, TB. But you're saying MAZ is different.
Oh, it's fundamentally different.
Yeah.
MAs is what we call an opportunistic pathogen. In humans, it's actually the leading cause of chronic pulmonary disease, especially for people with underlying conditions. But what our source material reveals is this stunning link between this, incredibly common bug and these rare recessively inherited immunodeficiencies in very specific dogs. It's a perfect, tragic storm of genetics and environment.
So our mission today is to dive into that precise intersection. We're going to unpack the tricky taxonomy of this bacterial family, figure out how these things survive in the environment, and most importantly, understand the shocking genetic flaw in breeds like the miniature Schnauzer that makes them so uniquely vulnerable.
And it all connects. We're linking environmental microbiology directly to canine precision medicine. And that ultimately has broader public health implications, what we call the One Health approach.
Okay, let's unpack this then. Let's start with what makes this whole family, the Mycobacteria, so different from, say, E. coli or staph.
Well, what defines them is their incredible resilience. It's all rooted in their cell structure. They're aerobic. They don't form spores. But the really practical difference is that they're acid fast.
Acid fast. Okay, that sounds technical. What does that actually mean for, like, a clinical lab?
It means they are incredibly durable. Their cell walls have these extremely high concentrations of mycolic acid, which is this waxy, lipid-rich layer.
So like a waxy armor.
Think of it exactly like that. It's a miniature, incredibly tough armored shell. And this armor, it stops standard techniques like the Gram stain from working. If you hit them with acid alcohol, which would kill most other bacteria, they hold on to their specialized stain.
So that lipid layer also makes them resistant to disinfectants, I'm guessing?
Precisely. They are survivors. And, while we're on the topic, it's worth noting that the scientific community is sort of fighting about what to even call them right now.
Oh, I found that fascinating. They want to redistribute 150 species from Mycobacterium into 5 completely new genera. Why? I mean, what's the benefit versus the cost of confusing everyone?
It all comes down to modern core genome sequencing. It gives us a much more precise understanding of their evolution and lineage. So the benefit is scientific accuracy, but the cost is real. Until everyone adopts the new names like mycobacteroids, it can definitely slow down information exchange between scientists and clinicians. So for clarity today, we're just going to stick to the traditional name, Mycobacterium.
Good idea. Okay, let's draw a clear line between the 2 main diseases this family causes. We've got tuberculosis, TB, and then mycobacteriosis.
Right. So the MTBC, the M tuberculosis complex, those are the major league players. They're obligate pathogens.
Meaning they have to live inside a host.
Exactly. And they have strong host preferences, like the human-adapted M tuberculosis. Dogs rarely maintain that kind of infection. They're usually just considered a spillover host from a human source.
So if TB is the major league human-adapted player, what are these opportunistic NTMs, the non-tuberculous ones?
That's our focus. NTMs they're everywhere. They're ubiquitous in soil, ubiquitous in water. And because they're environmental, they're opportunistic. They don't need a host to survive. And here's the key. Healthy humans, animals, are actually pretty resistant to them.
So it's when the immune system fails that the vulnerability appears.
That's it. Which brings us to M. avium, this big group of slow-growing NTMs. And the real villain in our story is one specific subspecies.
Which one is that?
It's M. avium subsp. lot hominisuus. Let's just call it MAH.
Yeah.
This is the critical player, especially in dogs. The sources we looked at propose that MAH is the only true environmental subspecies of M. avium.
And it's found in humans and swine, too.
Frequently. And it really does appear to be the main pathogenic one in dogs.
Okay, so MAH is the bug we're tracking. And we can officially dismiss that old idea that, contaminated chicken carcasses or bird droppings are a major source for dogs.
Absolutely. That's an outdated idea. MAH has only rarely been isolated from tuberculous lesions in birds. The real transmission is much more pervasive. It's linked directly to things like soil, groundwater, and tap water. The threat is all around them.
And this is where it gets really interesting for me. The environmental persistence. This bug is in our water systems, where it's exposed to things like chlorine. How does it survive that?
This is the amoeba angle, and it's just a perfect example of a successful evolutionary strategy. You have these free-living amoeba, which are common single-celled organisms in water systems, and they basically act as protective vehicles.
Like tiny biological submarines.
That's a great way to put it. Instead of becoming food, they become transport. Many of these mycobacteria are resistant to being eaten and killed by the amoebae. They get ingested, but instead of being destroyed, they just live unharmed and actually multiply inside the amoebal cyst.
Wow.
And that persistence helps MSC survive all sorts of adverse conditions, including chlorine. The amoebal cysts are effectively armored delivery vehicles.
That is just incredible. So for dogs, the infection route is typically breathing it in or drinking it from these sources that are everywhere.
Which brings us right back to the central mystery. If MAH is in the water bowl in the backyard, why are clinical infections so incredibly rare? And why are they so concentrated in just a couple of breeds?
Yeah, the numbers are stark. Only 43 published cases cited in the sources.
Which really emphasizes how strong a healthy canine immune system is. But when you look at who those 43 cases are, the breed predisposition is, frankly, terrifying.
It's undeniable. Miniature Schnauzers, 16 of those cases. Basset Hounds, 10 cases. That's more than 60% of all published reports in just 2 breeds.
It tells you right away the vulnerability isn't environmental, it's genetic.
And for the miniature Schnauzer, they've actually pinpointed the flaw. It's a tragic precision medicine success story.
It is. Researchers found a single inherited defect in an adaptor protein called CAR8D9. It's autosomal recessive, and it's been proven to be responsible for this massive susceptibility in Schnauzers.
Whoa, hold on. Autosomal recessive. Can you break that down for those of us who might have skipped genetics class? What does that mean for the dog?
It means the gene isn't linked to sex. And to get the disease, the dog has to inherit 2 defective copies of that gene, one from its mother and one from its father.
So if it only gets 1 copy.
It's perfectly healthy, but it's an asymptomatic carrier. And that's how the defective gene persists in the breed population. It hides.
And this protein, CARD9, what is its job in a healthy dog? What's going wrong?
Think of CARD9 as a crucial signaling protein. It's like a security guard for the immune system, essential for organizing the defense against fungi and, critically here, against intracellular bacteria like Mycobacterium.
Okay.
When a dog is homozygous for the variant, meaning it has 2 bad copies of the gene, that security system just collapses. The body can't mount an organized attack to kill the MAH bacteria that it inevitably encounters.
The source material was chillingly precise on this. It said miniature Schnauzers homozygous for this CARD9 variant always developed systemic mycobacteriosis.
It's a direct, almost one-to-one link. A complete failure of the innate immune response when exposed to a bug that almost every other dog just shakes off.
And what about the Basset Hound? Is it the same story?
Well, yes and no. They also show this unique susceptibility. The disease looks the same, but the specific genetic cause for them still needs to be found. We know the flaws there, but we haven't mapped the sequence yet.
Regardless of the specific defect, the clinical profile sounds devastating.
It is, and most of these dogs are heartbreakingly young, usually within their first 3 years. The symptoms, they start out really nonspecific, just some weight loss, lethargy, not eating much.
But it progresses quickly.
Very. Within weeks or months, it moves to severe generalized lymphadenopathy and splenomegaly, which means the infection is spread everywhere, to the spleen, the liver, the bone marrow.
That sounds like it would be incredibly difficult for a vet to diagnose early on.
Very challenging, yeah. If there aren't obvious skin lesions, the vet has to rely on biopsies or aspirations. They might see the acid-fast bacteria under a microscope, but the only way to know for sure which species it is involves cultures or expensive molecular methods. And we know the standard tuberculin skin test is completely unreliable in dogs.
So once you finally get that diagnosis disseminated MAC in a Schnauzer, what's the prognosis? The research says treatment is often discouraged, which is just a brutal thing to read.
It is the brutal reality of this disease. Because of that underlying insurmountable immunodeficiency from the CAR9 defect, treatment for the systemic form is generally ineffective. The disease is just chronic, progressive, and typically fatal no matter what you try.
Is there ever a good outcome for mycobacteriosis in dogs?
Only really in cases where the infection is localized, like on the skin, and the dog is otherwise immunocompetent. For those cases, in healthy breeds, a long course of triple drug therapy, usually rifampicin, clarithromycin, and fluoroquinolone, can work. But for the systemic genetic cases, the prognosis is grim.
And this leads us straight to the One Health idea, the public health risk. You have a pet with a systemic infection of a bacterium that we know causes lung disease in immunocompromised people.
Exactly. And while you probably won't catch it directly from the dog like a cold, the real risk is the dramatically increased exposure pressure. An infected dog is shedding and carrying vastly more MAH into the home environment.
So they're basically concentrating the pathogen in the house, raising the risk for anyone vulnerable.
Yes. An owner might be perfectly healthy, but if they're on immunosuppressant drugs or have an underlying condition, the risk goes way up. The source material is very clear. Any immunologically compromised people in close contact with an NTM-infected dog should be referred to their physician.
Okay, let's end on the positive side here, prevention. The discovery of that CARD9 defect is a massive victory, isn't it? It lets us intervene.
This is where precision medicine truly shines. Because the flaw in miniature Schnauzers is so well defined, we can predict susceptibility to this fatal infection before the dog ever gets sick. With a simple PCR test, you can test for homozygosity of that variant.
So you can predict this lifelong fatal risk before a puppy is even sold.
You can. And this rapid genotyping allows for the precise detection of dogs that will get sick and, just as critically, the identification of the asymptomatic carriers, the dogs with one copy of the bad gene.
Which lets breeders screen their dogs and stop passing it on.
It lets them eliminate this deleterious trait from the lineage. It stops the tragic cycle.
It really just highlights the need for collaboration between researchers, vets, and breeders.
That's the One Health approach in action. This genetic knowledge doesn't just save dogs. It gives us insights that go far beyond canine health.
We've covered a remarkable journey today. You have one of the most common bacteria on the planet, but it's only deadly when it's paired with this very specific hidden immune defect, like the CARD9 variant in Schnauzers, and the power of genetic screening is now our best tool against it.
And that knowledge has real promise for human medicine, too. The sources suggest that these CARD9 deficient miniature Schnauzers could be an invaluable animal model for studying host microbe interactions and even for testing new treatments for mycobacteriosis.
Which raises an important question. If we can isolate and study the precise molecular mechanisms of vulnerability in these dogs, what new doors might that open for treating acquired immunodeficiencies in human patients? Like transplant recipients or HIV patients who are also highly susceptible to this same opportunistic pathogen. That's something for you to mull over on your own.